In 7.3.2, we need to outline the structure of a ribosome. And when we do that, we need to talk about a couple of things. Talk about what the ribosome is made of. So it's made of protein and RNA. Also, it consists of two pieces, a big, large subunit and a smaller subunit. We have to mention the three binding sites, 1, 2, 3. And there's this little spot down here called the mRNA binding site. So basically, I just explained it, but let's just talk about maybe some specifics about this. So let me erase a little bit. So ribosomes are made up of a type of RNA called rRNA, and the R stands for ribosomal. And it's RNA. It's a single strand of a nucleic acid, of our nucleotides, right? Just like messenger RNA. The only difference here is that it has proteins like mixed in here. So if I'm just going to draw some squigglies, so pretend this is my single strand of messenger RNA. There are proteins embedded inside here that kind of hold it together. So it has this weird shape, similar to maybe like what you would think an enzyme, where it has that three-dimensional shape. So that's the rRNA part of it with the proteins. Now, as I mentioned before, there's a large subunit and a smaller subunit. What I want to say about this is that the RNA, oh my goodness, I'm sorry, that the ribosome doesn't sit like this in the cytoplasm or attached to the endoplasmic reticulum. It actually only has one half and the other half kind of like not really associated with each other yet. And they don't become associated until the messenger RNA, this pink molecule here, until the messenger RNA binds to this messenger RNA binding site here on the bottom. So once that happens, we only have the small subunit and the messenger RNA, and when that happens, the large subunit will plop on top. And the last thing I want to talk about is just the sites for the transfer RNA. So as review in translation, you're all experts now, but transfer RNA, which has this weird clover shape and a little thing on the bottom and another little arm down here. And we just talked about the acceptor stem, and on the top, we have a beautiful amino acid attached thanks to an activating enzyme. So that transfer RNA will come into this A site first, and the A actually stands for amino. So the A site first, because it's carrying that amino acid site. And then as our transfer RNA is moving from 5' to 3', this transfer RNA is going to move to the P site. Two things happen. It allows room for a second transfer RNA molecule to come in, but we begin putting together our amino acids to create our polypeptide. That is what the P stands for. So that's our polypeptide site. And at the end, it moves over one more time. The transfer RNA is amino acid-less. It doesn't have its amino acid anymore. And it leaves and it goes and looks for another amino acid. This E stands for the exit site. So amino site, polypeptide site, exit site spells ape if you're going backwards. And those are the transfer RNA sites. So that's all you really need to know about a ribosome. It's made of ribosomal RNA that has some proteins in it. There's a large and a small subunit, a place for messenger RNA to bind, and our three sites for our transfer RNA.